Welcome to all students in our ROSE project conducted by Rayat Shikshan Samstha, Satara. I am Mrs. Shinde S.R. from Sadhguru Gadge Maharaj College, Karad. We are going to study the topic biomolecules. In biomolecules, today we are going to study about the proteins. Now see the introduction. These proteins are the fundamental structural material of animal bodies. The proteins in the form of enzymes. Proteins are in the form of enzymes and which plays the role in the all physiological reaction. That means our body, there are the large number of physical reaction take place, and these reactions are take place with the help of enzymes, and these proteins are in the form of enzymes. The nutritional sources of proteins are milk, pulses, nuts, fish, meat, etc. Then see what are the proteins. The proteins are polyamides, which are high molecular weight polymers of monomers units called as alpha amino acids. That means polyamides. Poly means number of amide linkage are present in the proteins. Therefore, proteins are the polyamides which having the high molecular weight. The proteins are polyamides which having high molecular weight, and these are the polymers of alpha amino acids which are the monomers. Alpha amino acids are the monomers. Therefore, the proteins are polyamides of polymers or polymers of the monomer units which are called as the alpha amino acids. That means proteins are the polymer of the alpha amino acids, in which alpha amino acids are the monomer and proteins are the polymer. Now see the types of proteins. There are the two types of proteins. The first is the globular proteins. Then the globular proteins are the spherical in shape. Then it consists of the coiled and folded polypeptide chains. We know that the proteins are the polypeptides, and the globular proteins which consist of the coiled and folded polypeptide chains. The globular proteins are soluble in water. For example, insulin, egg albumin, serum albumin, and legumelin. This the second type of the proteins are the fibrous proteins. These are elongated rod-like shape. They consist of the long parallel polypeptide chains. They having the parallel polypeptide chains, and therefore it is the fibrous in structure. They are insoluble in water. For example, keratin and myosin. These are the types of proteins which are the globular proteins and fibrous protein. They are classified according to their shape of the proteins. The globular proteins having the coiled or folded polypeptide chains, and they are fibrous proteins having the long parallel polypeptide chains. They are soluble in waters, and fibrous proteins are insoluble in waters. Now see the alpha amino acids. We know that the polymers are the proteins are the polymer of alpha amino acids. Now see the alpha amino acids. This is the alpha amino acids. The alpha amino acids are the carboxylic acid. See here, this is the carboxylic acid, and they are the alpha amino acids. It are the alpha amino acids. That means the amino group is attached to the alpha carbon atom. This is the alpha carbon atom. The carboxyl group, and alpha carbon is the next to the carboxylic group. The carbon atom next to the carboxylic group. This is the alpha carbon atom, and this amino group is attached to this alpha carbon atom. And in the alpha amino acids, they having the four different groups. See here, this carbon atom having this carboxylic group. Then this is the R, it is the side chain. Then this is the hydrogen atom, and fourth group is this amino group. That means this carbon atom having the 4 different groups, and therefore this is the chiral carbon atom. This is the chiral carbon atom. Therefore, it is the optically active. Alpha amino acids having the chiral carbon atoms, except in glycine. This is the structure of glycine. In glycine, this carbon atom having the 2 hydrogen atoms. This carbon atom having the 2 hydrogen atoms. Therefore, this glycine does not having the chiral carbon atom. It is optically inactive. In this way, the alpha amino acids, except glycine, alpha amino acids are the optically active because they having the 1 chiral carbon atom. This is the R group side chain. This is the carboxylic group. This is the amino group. Next to the carboxylic group, it is the alpha carbon atom. And NH2, that means amino group, is attached to this carbon atom, alpha carbon atom. Therefore, it is called as the alpha amino acids. And this is the structure of the glycine. In glycine, there is the no R group. There is the presence of hydrogen atom. Therefore, it is optically inactive. Now see the classification of alpha amino acids. First, we see the structure, Fischer projection formula of this alpha amino acids, and it is the L form. It is the L form because the NH2 group is present on the left side. This is the carbon atom, chiral carbon atom. It is attached to the 4 different groups, COOH, carboxyl group, hydrogen atom, and this is the side chain R group, and it is the amino group. And this amino group on the left side, therefore it is called as the L form. And this structure is the L alpha amino acids. We know that the proteins are the polymer of these alpha amino acids, and on hydrolysis, the proteins gives the alpha amino acids, and which are in the L form in this structure. On the hydrolysis of protein, it gives the alpha amino acids which are in L form, called as the L alpha amino acids. These amino acids containing this side chain R group, and according to this R group, they are classified as acidic amino acids. Classified as acidic amino acids. That means it contains the R group as carboxylic group. The R group is the carboxyl group, and therefore this type of the amino acids are called as the acidic amino acids. That means the amino acids in which this R group is the having the carboxyl group. This side chain having the another functional group, and according to this functional group, they are classified as the first acidic amino acids. That means they are R group containing the carboxyl group. Then basic amino acids. They are classified as basic amino acids. In this basic amino acids, the R group contains amino group. The R group contains the amino group. Therefore, it is the basic amino acids. And third type is the neutral amino acids. The neutral amino acids in which the R group contains the neutral group. Neutral. R groups are neutral, or there is no any functional group. Therefore, this type of the amino acids are called as the neutral amino acids. That means the according to the presence of functional group in this side chain, the alpha amino acids are classified as acidic amino acids, basic amino acids, and neutral amino acids. Now see again they are classified as essential amino acids. What are the essential amino acids? These amino acids cannot be synthesized in human body and obtained through the diet. This type of the amino acids does not synthesized in human body. They are obtained from the diet. For example, valine, leucine, isoleucine, threonine, then methionine, phenylalanine, tryptophan, lysine, arginine, and histidine. These are the essential amino acids which are obtained from diet, because this type of amino acids does not synthesize in our human body. The next type is the non-essential amino acids. These non-essential amino acids are synthesized in human body. For example, glycine, alanine, asparagine, glutamine, serine, cysteine, tyrosine, proline, aspartic acid, and glutamic acids. These type of the amino acids are non-essential amino acids because they are synthesized in our human body. From the synthesis, they are classified as the essential amino acids and non-essential amino acids. Essential amino acid does not synthesize in human body, and non-essential amino acids are synthesized in our human body. Now see in this table there are the different types of the amino acids. These are the neutral amino acids, then acidic amino acids, and basic amino acids. We know that the neutral amino acids, that means the R group containing neutral group. These are the R groups which are present in these amino acids. These are the R groups. They does not containing the any functional group, or they all are in the neutral. Therefore, this type of the amino acids are called as the neutral alpha amino acids. Then acidic amino acids, they are containing the carboxylic groups. These are the aspartic acid and glutamic acid are the type of the acidic amino acid. Then basic amino acids, they containing the basic group. These are the basic amino acids. And also there is the essential and non-essential amino acids. These are the asterisk amino acids are the essential amino acids. In this way, they are classified as the neutral amino acids, acidic amino acids, and basic amino acids, also essential amino acids and non-essential amino acids. These amino acids are denoted by the symbol of first three letters. These are denoted by these first three letters. For example, this glycine, they are denoted as the GLY. Alanine, ALA, these are denoted by the ALA, first three letters. Also, it are represented as the first letter, only first letter. Generally, they are used as the symbol of the first three letters. In this way, the amino acids are represented by this symbol by using the first three letter. Now see the Zwitter ion. At a particular pH, the amino acid carries no net charge and is called as the Zwitter ion. Is called as the Zwitter ion. At particular pH, there is the no net charge. That means the pH at which the amino acid has a net charge of 0 is called as the isoelectric point. It's called as the isoelectric point, and at the isoelectric point, these amino acids form the Zwitter ion. These amino acids form the Zwitter ion. And at the isoelectric point, the positive and negative charges are equal. At the isoelectric point, the positive and negative charges are the equal, and at that point the charges net charge is the 0, and this position, this type of the ion is called as the Zwitter ion. Now see what is the Zwitter ion. See here, this is the alpha amino acid. The alpha amino acid containing the carboxylic acid group, acidic group. Also, it containing the basic group. That means the alpha amino containing the acidic group as well as the basic group. and therefore at the isoelectronic point, the hydrogen atom from this acidic group is towards the basic group. In this way, this acidic group, the hydrogen atom is liberated towards here nitrogen group. Nitrogen atom accept the hydrogen atom from this acid, and this having the positive charge and it having the negative charge. In this way, there in this structure there is the formation of positive and negative ion in both. It is the dipolar structure and is called as the Zwitter ion. Now see, according to the pH, they form the different forms. When at the low pH, that means it is approximately at 2, then these amino acids forms in the cation. And when at the high pH, this is formed at the anion form. The amino acids are formed in the anion position, and at the pH approximately 6, then it forms the Zwitter ion. It forms the Zwitter ion. Both positive and negative charge is obtained in the amino acids and is called as the Zwitter ion. In this way, according to the pH, the amino acids form the different forms. Now see the formation of peptide bond. The proteins are the polypeptides linkage. Proteins having the peptide linkage. Then how the peptide linkage is formed in the alpha amino acids. These are the alpha amino acids which containing the amino group, side chain and this carboxylic group. When two alpha amino acids are linked together each other, the OH group from this carboxylic group of this molecule and H from this NH2 group from the another molecule, there is the liberation of this H2O and formation of this C double bond NH bond, which is called as the peptide bond. Which is called as the peptide bond with evolution of this water molecule. Peptide bond is formed between the two alpha amino acids by evolution of the H2O molecules, and they form the C double bond O N H, and this type of the linkage is called as the peptide bond. In this way, when the two molecules of amino acids are linked together, then it is called as the dipeptide. Then combination of the third group with this dipeptide, then it is called as the tripeptide. In this way, the combination of the fourth, fifth and sixth molecules, then it is called as the tetrapeptides, then pentapeptides and hexapeptides respectively. When the number of amino acids linked by peptide bonds is more than 10, then this product is called as the polypeptides. These molecules are more than 10 molecules of the alpha amino acids, then it is called as the polypeptides. And the group, this CHR group, are linked together. Here CHR group, here is also CHR. These groups are linked together by this CONH, means peptide linkage, and this type of bonds are linked each other are called as the amino acid residues. These are called as the amino acid residues. And also in this polypeptide chain, when the molecules having the free NH2 group in this here, this is the dipeptide, here these molecules having the NH2 group free and is called as the N terminal, and the term end which find here free carboxylic acid is called as the C terminal end. In this way, the polypeptides having the two ends, N terminal and C terminal. N terminal on the left side and C terminal end on the right side. Now see the structure of the proteins. The protein structure of the proteins in the primary structure, secondary structure, tertiary structure, and quaternary structure. Now see the first primary structure of the proteins. In primary structures, they are made up of the long chains of amino acids by peptide bond. It is the sequence of constituent amino acids. It is the sequence of the constituent amino acid residues linked by peptide bonds. Linked by peptide bonds. The primary structure of the proteins is represented by writing the three letters. Represented by writing the three letters symbol of the alpha amino acid. See here, this one, they are writing the symbol in the three letters. Now these alpha amino acids are linked together with sequentially by peptide bond, and they are linked by in this way peptide bond. These are the amino acids monomer. These are the amino acids, and they are linked with each other by this, in this way, by peptide bond. In this way, in primary structures, the alpha amino acids linked with each other by peptide bond, and they form the long chain of the amino acids. Now see the secondary structure of protein. There are the two types of the structure, beta plated structure, and second is the alpha helix structure. These are according to the polypeptide chains linked with each other by the interhydrogen bonding. Interhydrogen bonding, that means there are the two separate polypeptide chains. This is the one polypeptide chain, and this is the second polypeptide chain, and they are linked together by this hydrogen bonding. That means the C double bond O, this oxygen atom is bonded with this NH. There is formation of the hydrogen bonding, and this is the interhalogen hydrogen bonding. This is the interhydrogen bonding which is present in the beta plated sheet-like structure. And in alpha helix structure, these polypeptide chains with intrahydrogen bonding. These polypeptide chains linked with each other by intrahydrogen bonding, and they form the alpha helix structure. Intrahydrogen bonding, that means the hydrogen bonding take place in the one polypeptide chain, and therefore they form the helix-like structure. Now see the tertiary structure of the protein. In tertiary structure, it is the final shape which is determined by the variety of bonding interaction between the side chains on the amino acids. That means in the tertiary structure, there is the bonding between the side chains take place, and this bonding between the side chains by the hydrogen bonds or ionic bonds or disulfide bridges or hydrophobic interaction. They are linked, the side chains are linked together each other by these hydrogen bonds or ionic bonds, disulfide bridges or hydrophobic interaction. And then see the quaternary structure. In quaternary structure, the spatial arrangement of these subunits of tertiary structures. That means these are tertiary structure, which are the subunits of the quaternary structures. That means these structures, there are the number of these type of the polypeptide chains, and they are linked together each other by and form this type of the structure is called as the quaternary structure. In this way, protein structure is studied in the primary structure, secondary structures, tertiary structure, and quaternary structure. Now see the denaturation of proteins. In denaturation, it is the process by which the molecular shape of protein changes without breaking the peptide bonds that form the primary structure. In denaturation process, the protein changes, they change their molecular shape without breaking the peptide bond. That means they are, after the denaturation of proteins, they are in the primary structure. This denaturation results in the disturbing the secondary, tertiary and quaternary structure of protein. They disturb the denaturation process. That means they disturb their secondary, tertiary, quaternary structure, and they are formed in the primary structure, and their shape is disturbing. And in this way, in denaturation process, they are formed in the primary structure. Denaturation causes the change in the properties of the proteins. See here, this is the structure of proteins, which is in the folded position. This one. And after denaturation, they are in the unfolded proteins. That means in the primary structure. After denaturation process, the proteins are formed in the primary structures, and this process is called as the denaturation.